Hello, it's Shudena and Garn. Today we're going to be solving May-June 2021, Paper 1, Variant 3. Let's begin. 1. Which set of measurements is correct? Diameter of a capillary. As we know, the diameter of the capillary is going to be 7 micrometers. The reason for that is that it's the same size as red blood cell, which is also going to be 7 micrometers. Then we have the thickness of cell surface membrane of red blood cell. The thickness of any cell surface membrane is always going to be 7 nanometers. So the answer is going to be A. The diagram shows an eyepiece graticule and part of stage micrometer scale as seen using 100 magnification, which is the correct method for calculating the value of one eyepiece graticule unit in micrometers. So the first step that we have to do is convert this millimeters into micrometers by multiplying by 1000. Therefore, multiply 0.1 by 1000 is correct. Then what we do is we divide the length of the micrometer scale by the amount of units on the eyepiece graticule. Here it's standardized, we have 100 units on the eyepiece graticule. So it's going to be 100 divided by 100. So the only correct option is divide by 100 only, then it's going to be C. 3. A prokaryotic cell, 1 micrometer in diameter, is magnified 50,000 times on an electron micrograph. What is the diameter as shown in the electron micrograph? Here it's asking us for the image size, so let's draw the equation for the magnification. How to calculate this is we multiply the actual size by magnification. Here the actual size, we have it in micrometers. So we multiply 1 by 50,000 gives us 50,000 micrometers. And here it wants the answer in millimeters. So to convert from micrometers to millimeters, we divide by 1000. So the answer is going to be C. 4. Which cell structures contain DNA? Let's start with mitochondria. It definitely does contain DNA, but it's circular DNA. So this is correct. 2. Chloroplasts. It also contains circular DNA. Centrioles. Centrioles do not contain DNA because centrioles are made of 2 microtubules lying 90 degrees to each other or at right angles. They are found at the poles of the cell during mitosis, and they produce the spindle fiber also during mitosis. So this is incorrect. For 4, nucleolus. Yes, it definitely does contain DNA. So the answer is going to be B. Number 5. 4 students were asked to match the function with the appearance of some cell structures in an animal cell. The functions were listed by number. To start with, mRNA passes through to the ribosome. We all know that when mRNA is formed during transcription, it passes outside the nucleus through the nuclear envelope, through the nuclear pores. Synthesis of polypeptides. We all know that polypeptides are synthesized in the ribosomes. Next, synthesis of lipids. We all know lipids are made in the smooth endoplasmic reticulum. Let's see the suggestions. Here for 1, it's either V or X. Let's see V. Membranes which surround an enclosed inner cavity. This is incorrect because this is not the nuclear envelope. For X, a double membrane interspersed with pores. This is correct, and it does have pores, so 1 is going to be X. For 2, synthesis of polypeptides. We have either W or Z. For W, non-membrane bound spherical structures. This is correct because we all know ribosomes are spherical and they do not contain a membrane. For Z, it's membrane-bound sacs arranged as flattened stack. Flattened stack is incorrect because this is the Golgi body. So the correct answer is going to be W. For V, both of them are the same answers. Membranes, which surround an enclosed inner cavity. This is the smooth endoplasmic reticulum. So the answer is going to be C. Question number 6. Which row is correct for structures found in eukaryotic cells? To start with, circular DNA. This is correct in case if it's an animal cell, it's only found in the mitochondria. If it's a plant cell, then it's only found in the mitochondria and the chloroplasts. So it's correct, and both of them are eukaryotic cells. So it's present 70 S ribosomes. As we said, they are present in mitochondria if it's an animal cell, and present in both chloroplasts and mitochondria if it's a plant cell. 80 S ribosomes, they are present in the nucleus and the cytoplasm. So the answer is going to be A. 7. 4 solutions were tested for the presence of 4 different biological molecules. The appearance of the solution after each test are shown in the table. Which solutions contain molecules with ester bonds? Out of all these samples, we know that fats are going to be the ones which contain the ester bonds. The reason for this is to form a triglyceride, a glycerol joins with 3 fatty acids by ester bonds, releasing 3 molecules of water in a condensation reaction. Therefore, an ester bond is formed between the hydroxyl group of the glycerol and the carboxylic acid group of the fatty acid. Now, to the second point, a positive test for fats is a cloudy emulsion after the test. So the only cloudy options we have is 1 and 3, so the answer is going to be B. 8. The diagrams represent 2 monosaccharides with the same molecular formula. Both can exist in alpha or beta form as shown. The diagram shows lactose molecules formed by condensation between glucose and galactose. Which molecules have condensed to form lactose? Let's start with this first one. As you can see here, it's definitely going to be beta-galactose. The reason for this is because we have the hydroxyl group facing upwards. And for the second one, it's definitely also going to be beta-glucose, because the hydroxyl group here is facing upwards and here it's facing downwards. By the way, you could get confused thinking that the second monomer is rotated like cellulose, but this is incorrect. So the answer is going to be beta-galactose and beta-glucose. So it's going to be D. 9. What type of covalent bond between 2 monomers is shown? Which molecules contain this type of covalent bond? So if you haven't noticed yet, a bond is formed between carbon 1 and carbon 6. So it's 1-6 glycosidic bond. And these are found in branched carbohydrates. For example, we have amylopectin and glycogen. Let's see the suggestions we have here. Amylopectin, amylase, glycogen, and starch. Amylase is incorrect because it only has 1-4 glycosidic bonds, not 1-6. B, amylopectin, amylase, this is also incorrect. C, amylopectin, glycogen, and starch only. All these 3 suggestions are correct. Amylopectin contains 1-6 glycosidic bonds. Same with glycogen. Starch is made by amylose and amylopectin. And as we said earlier, the amylopectin contains both 1-4 glycosidic bonds and 1-6 glycosidic bonds. Therefore, C is going to be correct. Which statements about carbohydrates and triglycerides are correct? 1. They form polymers. Only carbohydrates form polymers, not triglycerides. The reason for that is because polymers means giant molecules made by many repeating units. Triglycerides are only formed by combining glycerol and fatty acids, which we have mentioned earlier, so it's not a giant molecule. So 1 is incorrect. 2, they contain carbon, hydrogen, and oxygen. This is correct. Going back to the drawing here, we could see that glucose contains oxygen molecules. so 2 is correct. For 3, they are used as energy stores. This is correct for both carbohydrates and triglycerides, so the answer is going to be D. 11. Which molecules contain at least 3 double bonds? We all know that proteins have a carboxyl group, and this carboxyl group contains double bonds. So let's look for proteins. Saturated fatty acid, this is incorrect. B. Collagen and saturated fatty acid. Collagen is correct, but this is incorrect. C. Hemoglobin and collagen. Because these are both proteins, C is the correct answer. 12. The diagrams show the structures of 2 amino acids. One contains 2 amino groups, labeled 1 and 2. The other contains 2 carboxylic groups, labeled 3 and 4. A peptide bond is formed between the 2 amino acids. Which groups form the peptide bond? So you have to pay attention to something. Here is the R group. It's not the amine group, this is the amine group. And peptide bonds are formed between amine groups and carboxylic acid groups, not the R groups. So we know that 1 and 4 is going to be incorrect. The answer is B. Which row about the structure of proteins is correct? Let's start with the primary structure. Is the number of amino acids present in a protein? This is correct. It's basically the sequence of amino acids in a polypeptide. B. Is the order of amino acids present in a protein encoded by the DNA? This is correct. As we said, it's the order of amino acids. C is the result of translation of an mRNA molecule by a ribosome into a chain of amino acids. This is also correct. D is the sequence of amino acids. This is also correct. And in a primary structure, the only peptide bonds are formed. Secondary structure. It's the hydrogen bonding between the oxygen in the carboxyl group and the hydrogen in the amine group. So let's start with A. Is the right-handed spiral formed by the primary structure? By means the right-handed spiral, it means the alpha helix. So this is correct. B is the coiling of a chain of amino acids to form B pleated sheet or an alpha helix. This is incorrect because actually it can form both in the same polypeptide, so this is incorrect. C occurs because of an attraction between hydrogen and oxygen atoms in the peptide bond. This is correct. As we mentioned earlier, D is formed by hydrogen bonding between amino acids forming the primary structure. This is correct. Tertiary structure. Just a brief note, it's the bonds formed between the R groups or side chains of amino acids. It could be disulfide, ionic, hydrogen, and hydrophobic. Let's see the suggestions. Is the result of cross-bonding between specific amino acids in the primary structure. This is correct, it's between amino acids. B is the shape formed by the folding of a polypeptide and is held together by hydrogen bonds. Hydrogen bonds only is incorrect. As we said, there are 4 different bonds in the tertiary structure. C is the result of ionic and hydrogen bonds, disulfide, hydrophobic interactions between amino acids. This is correct. D is formed as a result of interaction of the side chains, aka R groups of amino acids in the primary structure. This is correct. And lastly, we have the quaternary structure. In the quaternary structure, these same bonds are formed, but between different polypeptides. In the tertiary structure, it's between the same polypeptide, but in the quaternary structure, it's between different polypeptides. Incorrect. B contains 2 types of polypeptide that interact, forming the shape of a protein. No, it's not two types of polypeptide. It could just be more than 1 polypeptide. It doesn't necessarily have to be 2 types, so this is incorrect. It's formed by 4 polypeptides. As we said, it's only more than 1, not a specific number. So this is incorrect. Lastly, formed by linking together of more than 1 polypeptide to form a protein. This is correct. The answer is going to be D. 14. The enzyme beta-galactosidase can catalyze the hydrolysis of 4 substrates with similar structures. Each substrate gives a different Km value, or Michael-Menton's constant value. For which substrate does beta-galactosidase have the highest affinity? So as we said, the Michael-Menton constant and affinity are inversely proportional. By means, if Michael-Menton constant increases, the affinity decreases. So here it's asking for the highest affinity, therefore the lowest Km value. So here the lowest Km value is going to be D. An investigation was carried out on the effect of temperature on the activity of an enzyme when it's immobilized and when it's non-immobilized, aka free in solution. The product of the enzyme-catalyzed reaction causes a decrease in pH. The graph shows the results of the investigation. By means, the more efficient the enzyme and the faster it catalyzes the reaction, the more decrease in pH per unit time, which would give the highest yield of product. So what we're looking for here is the most decrease in pH per time. And here we could see this line, which represents the non-immobilized at 37 degrees. So the answer is going to be C. An indicator mixed with agar forms a pink color. The pink-colored agar becomes colorless when put in acid. Blocks of pink-colored agar are cut to different sizes and put in acid. All other variables are kept constant. Which block becomes colorless more quickly? Here in this investigation, diffusion is investigated. So in diffusion, the faster the diffusion, the larger the surface area to volume ratio. Therefore, I've done the calculations previously. The highest surface area to volume ratio is going to be A, so the answer is A, as you see here. 17. 3 identical plant cells were put into one of 3 different concentrations of sugar solution, 10%, 5%, and 2.5%. The cells were left for 50 minutes and then observed using a light microscope. Which statement is not correct? For A, cell X has the same water potential as the sugar solution it was put into. This is correct. The reason for this is because water didn't go in, so it's turgid, and water didn't move out, making it plasmolized. For B, cell Y is turgid and cell Z is plasmolized. A turgid cell is when water enters the cell, aka inflaming it as a balloon, and Z is plasmolized is when water moves out and cell membrane constricts. So this is correct. C. Cell Y was put into a 2.5 sugar solution. This is correct. The reason for this is because it's the lowest sugar concentration out of all 3. By means, water will enter the cell by osmosis down water potential gradient, because outside is lower sucrose concentration. For cell Z had more negative water potential than the sugar solution it was put into. By means more negative water potential, so a lower water potential. If that was the case, then water will go inside the cell, making it turgid. But in this case, the cell is plasmolized, so this is the incorrect answer, so the answer is going to be D. 18. How many copies of each different DNA molecule are found in a cell at the start of each of these stages of a mitotic cell cycle? So let's first make something clear. Here you could see the S phase. In the S phase, DNA replication takes place. By means, the amount of DNA is going to be multiplied by 2. Therefore, all the stages after that S phase up until the end of cytokinesis is going to contain 2 copies of DNA. So the appropriate answer is going to be D. 19. Hydra are simple animals which can reproduce asexually. This is an important point. The photomicrograph shows an adult hydra with new hydra developing while attached to the side of the adult animal. Which processes have occurred in the two hydra? 1. DNA replication. If it's dividing by mitosis to reproduce asexually, then definitely DNA replication would take place because it's part of the cell cycle. So this is correct. For 2, growth has taken place. You could clearly see the number of cells increased. So this is correct. 3. Mitosis. If growth and DNA replication took place, this is the features of mitosis, or that's the benefits of mitosis, then definitely it did take place. So the answer is going to be A. The diagram represents a nucleotide containing thymine. Which statements about this nucleotide are correct? 1. Thymine is a pyrimidine. This is correct because it's a single ring base, and pyrimidines is single rings. Double rings is going to be purines, and it's adenine and guanine. Pyrimidines is thymine and cytosine. So this is correct. 2. Base pairing occurs with 2 hydrogen bonds. This is correct. Thymine pairs with cytosine. 2. Base pairing occurs with 2 hydrogen bonds. This is correct. The reason for that is adenine pairs with thymine by 2 hydrogen bonds. Cytosine pairs with guanine by 3 hydrogen bonds. So this is correct. 3. The carbohydrate can be ribose or deoxyribose. So ribose is the nucleotide forming mRNA. Deoxyribose are the nucleotides forming DNA. As we all know, here it says containing thymine. mRNA or mRNA nucleotides do not contain thymine. They contain uracil instead of thymine, so it cannot be a ribose. It can only be a deoxyribose. So 3 is incorrect. The answer is going to be B. Some of the events that occur during transcription are listed. 1. Bonds break between complementary bases. Bonds form between complementary bases. Sugar phosphate bonds form, aka phosphodiester bond. Free nucleotides pair with complementary nucleotides before the mRNA molecule leaves the nucleus. Which events occur twice during transcription? Huge emphasis on twice. To start with, bonds break between complementary bases. This is correct, it does happen twice. The reason for this is the bonds break once between the DNA when it unzips, because as we all know, the double helix is held together by hydrogen bonds. So it breaks for the first time during the unwinding or the unzipping of DNA by the enzyme helicase. And bonds break for the second time when the complementary RNA nucleotides, as you can see here, binds to the DNA by hydrogen bonds. When that mRNA is complete, or when transcription is complete, the RNA or the mRNA or the hydrogen bonds formed between the DNA and the mRNA break, so the mRNA is able to leave the nucleus. So this is correct. I hope I made it clear when it happens the first time and when it happens the second time. 2. Bonds form between complementary bases. So the first time is when mRNA nucleotides binds to the DNA by hydrogen bonds via complementary base pairing. So it happens once here. And then the other time when transcription is complete and the DNA is binding back together via complementary base pairing. So this is correct. 3. Sugar phosphate bonds form. This is incorrect. It only forms once when mRNA is complete. 4. Free nucleotides pair with complementary nucleotides. This only happens once when mRNA nucleotides bind with the DNA, so this is incorrect. So the answer is going to be D. 22. The table shows the DNA triplet codes for some amino acids. 2 mutations occur in this sequence during DNA replication, which mutated DNA would result in a polypeptide with 1 different amino acids. First of all, to make things clear, we all should know that DNA code is degenerate. By means, several DNA triplets could code for the same amino acids as we see here. The same amino acid is coded for by different DNA triplets. Therefore, it's asking which would result in a polypeptide with 1 different amino acids. I've annotated it for you to make it easier. To start with A, here it's mutated to a stop codon, so this is incorrect. And here it's mutated to a triplet code with the same amino acid, so it did not change, so this is incorrect. Same thing with B. It mutated to the same amino acid. This is also incorrect. For C, as we can see here it's arginine and here it's glycine. So it mutated into a different amino acid. So this is correct. Here the same issue. It's mutated to the same amino acid too. So the answer is going to be C. 3. A gene codes for the sequence of amino acids in a single polypeptide. Hemoglobin consists of 2 alpha globins and 2 beta globins. How many genes are needed to code for a single hemoglobin molecule? If you put 4, then this is incorrect. The reason for this is you only need 1 copy of DNA to code for as many alpha globins or as many beta globins as you could possibly need. You only just need 1 DNA template, that's it. So you need 1 for coding for alpha globin and B coding for beta globin. So you only need 2, the answer is going to be B. 24. Which properties of water molecules are important in the upward flow of water through the xylem? 1. Water molecules are attracted to each other by hydrogen bonding. This is correct, and this is called cohesion. 2. Water molecules are attracted to cellulose by adhesion. This is correct, due to the hydrophilic properties of cellulose. 3. Water molecules have a high cohesion in water columns. As we said, this is correct. The reason for this is that water is a polar molecule. Polar by means there is a partial negative charge on the oxygen and partial positive charges on the hydrogens. Therefore, they are able to form hydrogen bonds with each other and with polar molecules. Therefore, the answer is going to be A. 5. The graph shows the rate of water absorption and the rate of water loss by a plant during one 24-hour period. The plant was grown in natural conditions. What may be concluded from the graph? 1. The rate of water absorption and the rate of water loss peak at 16. This is correct. 2. The rate of water loss is greater than the rate of water absorption for 12 hours. This is correct because overall, you can see that water loss peaks the most. So this is correct. 3. The rate of water absorption is greater than the rate of water loss at night. Let's see at night. It's here and here. So as we see, the rate of water absorption, and it's this line, is greater at night. So this is correct. The answer is going to be A. 6. Which statements explain why a stem is cut underwater and connected to a potometer underwater? 1. To prevent plasmolysis of xylem vessel elements. This is incorrect. Plasmolysis could happen only if the water potential of the exterior fluid is much lower than the water potential of the xylem. So this is incorrect. 2. To prevent the collapse of xylem vessel elements. Xylem vessel elements, if you cut them in air, not underwater, they will not collapse because they are hollow and lignified. So this is incorrect. 3. To prevent air entering the xylem vessel elements. This is correct. The reason for this is if we are here having water molecules attracted to each other by hydrogen bonding, aka cohesion, if a giant air bubble enters it basically disrupts that cohesion column. Therefore, we do not want any air bubbles entering because it will affect the rate of transpiration. So the correct answer is going to be D. 27. Sucrose moves into an actively dividing shoot tip from the phloem sieve tube element, which changes to the water potential and the volume of liquid in the phloem sieve tube elements are correct. So here, sucrose moved from the phloem sieve tube element into a cell. Therefore, what's going to happen is the water potential is going to become less negative because sucrose moved out. And the volume of liquid, if the sucrose moved out of this phloem sieve tube elements, it's normal for some water to also go out with it, so the volume of water decreases. Therefore, A is correct. Sucrose is loaded into phloem sieve tubes from companion cells. What is the correct order of statements that explains this mechanism? The first step in transporting sucrose is that companion cells, let's draw it here, pumps out hydrogen ions into its cell wall by proton pumps, by proteins in the membranes called proton pumps. So the first step is going to be 2. Then what happens is that hydrogen ions diffuse into companion cells through co-transporter proteins. When hydrogen ions are now out, there is a high proportion of sucrose outside the companion cell. So what happens is both the hydrogen and sucrose move back into that companion cell through a membrane protein called a co-transporter protein. Now, the hydrogen ions moves down concentration gradient and the sucrose moves against concentration gradient into the companion cell. So this is step number 2. Step number 3, as we said, it's sucrose co-transported along hydrogen ions. We just mentioned that, so this is going to be the third one. And then sucrose diffuses into phloem sieve tube elements via plasmodesmata. Once it's inside the companion cell, here we have sieve tube elements. The high proportion of sucrose inside the companion cells diffuse into the phloem sieve tube elements through the plasmodesmata and its strands of cytoplasm to the phloem sieve tube element. And this is the process of the loading of sucrose. So the answer is going to be C. 29. Which components are found in arteries? All 3 of these are found in arteries in high proportion. Collagen makes the arteries able to withstand the large pressures. Same as smooth muscle. Smooth muscle allows pressure to be modified by contracting or relaxing. So the answer is going to be B. Collagen, endothelium, and smooth muscle is correct for both arteries and veins. However, if we're talking about capillaries, capillaries only has endothelium. Question 30. Which diagram correctly shows the direction of flow of blood through the heart? Let's start with that first one. As we see here, blood is entering the atrium and going out of the ventricle. This part is correct. But here, the blood is entering through the ventricle. This is incorrect. For B, as we see here, blood is entering to the atria, then to the ventricle, then out, and here it's entering to the atria and then to the ventricle. So this is correct. The answer is going to be B. Let's see the rest. This is incorrect. This arrow is incorrect. It's supposed to enter through the atria, same as with D. So the answer is going to be B. 31. Which row correctly identifies the pulmonary artery? To make things clear, we have to first know whether the pulmonary artery has deoxygenated or oxygenated blood. As we all know, pulmonary artery goes to the lungs, from the ventricle to the lungs. Why? So hemoglobin is able to to bind with oxygen, forming oxyhemoglobin in the lungs. Therefore, this basically means that the blood in the pulmonary artery is definitely going to be deoxygenated. So this is correct. And as we know, because it's an artery, the thickness of the blood vessel wall is definitely going to be thicker. So this is correct. And because it's also an artery, we know it's going to be at a higher pressure than 3 to 8. So this is going to be correct. The answer is A. Found in all blood vessels, lymph, and tissue fluid. Carbon dioxide, this is correct. It's a waste material found in all 4. 2. Glucose, this is correct. It's required for respiration. 3. White blood cells, this is correct. Same with antibodies. So the answer is going to be A. And by the way, lymph has a very similar composition to tissue fluid. 33. At high altitudes, the oxygen content of the air may be a third of that at sea level. So there's a lower partial pressure of oxygen. As a person slowly climbs a mountain, their body gradually adjusts to the high altitude. What is increased during this period of adjustment? A. The concentration of hemoglobin in red blood cells. This is incorrect. The concentration of hemoglobin does not increase. B. The oxygen carrying capacity of hemoglobin. This is incorrect because the affinity does not change at all. Let's write here, affinity is not altered. C. The number of red blood cells per millimeter cubed of blood increases. This is correct. The reason for that is to compensate for the lower partial pressure of oxygen at a higher altitude, so this is correct. D. The rate at which hemoglobin releases oxygen into tissues. This is incorrect because that rate depends on the concentration of carbon dioxide, has nothing to do with altitude. So the answer is going to be C. For which row correctly shows features present in terminal bronchioles? As we all know, bronchioles do not have cartilage, so this is correct. Trachea has C-shaped cartilage, and bronchioles has irregular blocks of cartilage. And bronchioles do have cilia and do have smooth muscle for support, so the answer is going to be C. The photomicrograph shows a section through lung tissue. Which structures are present in this photomicrograph? The first thing my eye must come on is the irregular blocks of cartilage. Because as we have mentioned earlier, bronchus do have irregular blocks of cartilage, so definitely bronchus is present. Point number 2, here we could see a regular shaped structure. This might be an artery. And here we could see an irregular shaped structure too. So this might be a vein. Moreover, for the trachea, as we mentioned also earlier, it has C-shaped cartilage. And here I cannot see any C-shaped cartilage. So this must be the only correct solution. So the answer is going to be A. The symptoms of 2 diseases are listed. Before we start answering this question, the syllabus for 2022 onwards has this part deleted, so it's not in the syllabus anymore, but we're going to answer just in case. To start with, which row identifies diseases 1 and 2? Disease 1, coughing up blood, pain when breathing, loss of weight. Let's just ignore this for the moment. Disease 2, shortness of breath, difficulty breathing out, fatigue. This is definitely going to be emphysema. The reason for that, shortness of breath, is because the surface area to volume ratio of the lung decreases because the alveoli bursts. So less oxygen is going to be absorbed into the body. So emphysema is correct. For disease number 1, it emphasizes here pain when breathing, and this distinguishes lung cancer from chronic bronchitis. So lung cancer is going to be the appropriate choice. So the answer is going to be D. 37. Disease transmission can be reduced in different ways. Antibiotic therapy, vaccination, more living space per person. The transmission of which disease can be reduced by all these methods. Let's start. Antibiotic therapy means that this is definitely going to be a bacterium. Vaccination for non-sufferers. It protects against all of these. More living space per person. Here is the hint. More living space per person means that this is an airborne disease. The airborne diseases that we have here, it's either measles, which is a virus, or tuberculosis, which is a bacterium. But because here it mentions antibiotic therapy, antibiotic only works against bacteria, not viruses. So the appropriate answer is going to be B. Number 39. The diagram shows the immune response following infection by a virus. Which row identifies the cells labeled? Let's start with the most easier and obvious one. For G, we can see that a cell is attached to a host cell infected with a virus and almost looks like it's going to kill it in some way or shape or form. This is definitely cytotoxic or killer T cells. These cells attach to the host cell infected with virus that shows specific antigen and secretes a substance called perforin. This substance kills the infected cells by inducing something called apoptosis, which basically means self-digestion. We know that G is going to be T killer cell. Coming to F, here we could see the phrase chemical stimulation, which means that this must be a T helper cell, because T helper cells secrete cytokines. These are chemicals which activate the B cells, which is shown here. So we know that F is going to be T helper cell, and E must be a B memory cell, because B cells divide into both memory cells and plasma cells. So the answer is going to be A. 40. Monoclonal antibodies are used to test for the presence of the hormone HCG in the urine of a human female during early pregnancy. By the way, just a side note, HCG stands for human chorionic gonadotropin. Which statements describe how the monoclonal antibodies used in this test are produced? 1. HCG is injected into a mouse and plasma cells in the mouse produce antibodies specific to HCG. This is correct because plasma cells secrete antibodies. This is correct. 2. Antibodies are extracted from the mouse and then fused with cancer cells to produce hybridoma cells. Antibodies is incorrect. It's supposed to be antigens. Antigens is what stimulates an immune response in the mammal, not the antibodies. So this is incorrect. 3. Single hybridoma cells are cultured and they divide by mitosis to produce a clone of hybridoma cells. This is correct. So hybridoma cells is when plasma cells fuse with myeloma cells, aka cancer cells, by a substance called a fusogen, forming hybridoma cells. And these cells, because they contain myeloma, aka cancer cells, we know that they divide uncontrollably. Therefore, they are able to divide by mitosis. So the answer is going to be C. Thank you very much for watching this video. If you think this channel is useful, please share and subscribe and like this video.